we just spent some time thinking about mitosis, so the process by which cells can divide and create identical replications of themselves. So you can have, one parent cell creating two identical daughter cells. So if this was, a cancer cell, it would create two identical cancer cells that have the same genetic information inside of them that the parent cell had. Or if this was a skin cell, it would create two identical skin cells with the same genetic information inside of themselves that the parent cell had. The other type of cell division I want to talk about is meiosis. And so both of these are cell division. And both of these happen at the end of the cell cycle. And so the cell cycle, remember, is just the life cycle of a cell, and most cells are going to be doing this for their whole life, always, no matter what, mitosis. But some cells, a very small amount of tissue in our bodies, does meiosis instead at the end of its cell cycle. And the cells that do that in us are known as oocytes and spermatocytes. And you can probably guess by the name of the second one exactly what these cells are going to be making. Yeah, so spermatocytes, they make sperm, and oocytes, they make eggs. Cytes here just means cell. So an egg-producing cell or a sperm-producing cell. And the way that meiosis is different from mitosis is you're still starting out with a parent cell that has all the genetic information from the host organism. So in us, our oocytes and our spermatocytes, they have two sets of 23 chromosomes, equaling 46. But at the end of meiosis, instead of having exact copies like we did in mitosis, at the end of meiosis, we now have sperm or eggs that have one set of 23 chromosomes inside of them. And this way, you can think of a sperm meeting an egg, each of them carrying 23 chromosomes, coming together to fertilize, to create a zygote, a fertilized egg, that has 46 chromosomes, two sets of 23. So sperm and egg each carry half the information to create a human being. And so the process of meiosis allows our bodies to go from a full set of information, two sets of 23, to only one set of information, one set of 23. And so the daughter cells are going to be different than the parent cell. Daughter cells have half as much DNA as the parent cell did, but critically, each of the daughter cells is different from each other. In mitosis, the daughter cells were identical to the parent and to each other. In meiosis, the daughter cells are different from the parent and different from each other. So this process creates genetic diversity in individual species. And so we'll talk a little bit more about how that works. The secret to DNA replication and the steps of division in our cells is that it's exactly the same in both processes. It's the same in mitosis versus meiosis, but the only things that are different are how many times the cell splits in two, how much DNA ends up in each cell, and how the DNA aligns during division. So if we look at that first one there, how many times the cell splits in two. In mitosis, we only divide once, so we started with one parent cell and we're ending with two daughter cells. In meiosis, the cell splits in two twice. So we still start with one parent cell, but then we divide into two, and then those two divide again into four. And so we have two steps of division in meiosis, whereas we had only one step of division in mitosis. And then how much DNA ends up in each cell. Again, mitosis, you're making an exact copy. So you started with two sets of 23 chromosomes, and you're ending with two sets of 23 chromosomes. But in meiosis, you're starting with two sets of 23, and you're ending with one set of 23 in that one, in that one, in that one, and in that one. And so compared from start to finish, you've halved the amount of genetic information in the cells from the beginning to the end. And then lastly, the only other way that they differ is how DNA aligns during division. So remember the steps of mitosis, PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. We're still going to use the same verbs here. We're still going to think about, attaching, aligning, pulling, and dividing. But something special is going to happen during that alignment part of meiosis, and meiosis 1 specifically. In this very first couple of steps of meiosis, we're going to have what's called crossover. And so I want to talk a little bit more about that. So if we go ahead and think about this first half of meiosis, we're still going through all of these, verbs. We're still going through all of that PMAT. But the homologous chromosomes are going to cross over. And when they cross over, they're mixing and segregating themselves. And so at the beginning, we had information from mom's chromosomes and information from dad's chromosomes making exact copies of themselves into sister chromatids. But during alignment, look at those little chromatid arms. Look at the arms of those sister chromatids and how they are overlapping with each other. So the homologous pair here from mom and dad is overlapping. The homologous pair here from mom and dad is overlapping. Excuse me. And when they overlap, they can exchange information. And so if we look at what's happened after they start to pull apart here, now we've got a little bit of information from dad's chromosome on mom's, a little bit of information from mom's on dad's, and so on and so forth. And let's zoom in a little bit more at exactly how this is working. So we call this crossover, it's the process of synapsis or fusion, where the sister chromatid arms come together and exchange information. And so if we think about my textbook example from, I think we talked about that in lecture 4, you can imagine that all the information in your cells is a textbook on how to build a human. And you've got 2 copies of that textbook. You got a copy from mom with all the information for how to build a human, and you got a copy from dad with all the information for how to build a human. And in that book, there are 23 chapters. And so chapter 1, chromosome 1, here's information on how to do a whole bunch of things, like, neuroblasts. Chapter 11, chromosome 11, here's information on how to build hemoglobin. Chapter X, chromosome X, here's information on red blood cells, all sorts of things. Or, chapter 6, chromosome 6, here's information on how to make hair. In mom's book, it might say make brown hair. In dad's book, it might say make blonde hair. And so each chapter holds the same sorts of information, but slightly different variations on that information, slightly different variations on those traits. And so this process of crossover allows us to mix up that information. So you can imagine we started with a textbook from mom and a textbook from dad on how to build a human. How to build a human. It's an H. And then during the process of crossover, you're going to take some pages out of mom's book and put them into dad's book. You're going to take some pages out of dad's book and put them into mom's book. And so instead of having 2 distinct sets of information on how to build a human, one that came from your mom's egg, one that came from your dad's sperm. Now you're going to take all the information that you have in your 2 textbooks and mix them together. And so maybe in meiosis from one oocyte, you took page 67 out of mom's book and put it in the place of 67 in dad's book, and took 67 from dad's book and put it in the place of 67 from mom's book. Maybe in a different process of meiosis, you took pages 100 through 150 and swapped those between the 2 books. And every single time that you do this, you're swapping a slightly different amount of information between the 2 books, and slightly different pages and slightly different information from each chromosome. Remember, every chromosome has hundreds to thousands of genes on it, and so you can switch a couple of those genes, you can switch hundreds of those genes, you can switch one of those genes, but if you swap any information, you're now making one chromosome different from another. And so if we look at this specific example that we have here, this set of sister chromatids are exact copies of each other. And so you've got, 3 dominant alleles here, 3 capital A, B, and C. These sister chromatids are exact copies of each other. You've got 3 recessive alleles here, lowercase a, b, and c. If we just did mitosis at the end of cell division, we would have these exact same sets present in the daughter cells. But what happens in meiosis is because we're going to get some changeover, because we're going to swap some pages between mom's information and dad's information, now we're going to end up with 4 chromatids. We can imagine we would cut these down the middle at the end of meiosis. We're going to end up with 4 chromatids, one that looks the same as the original mom, one that's got capital ABC, but this one over here, it's got a different mixture, a new mixture that we didn't see over here, capital A, capital B, lowercase c. Another new mixture here, lowercase a, lowercase b, capital C. And then another copy of the original information, ABC here. And every single time meiosis takes place, we're doing a slightly different exchange of alleles across 2 chromosomes. And so you're mixing up the information to create new diversity, to make every sperm different than every other sperm, to make every egg different than every other egg. This is allowing for 2 people to have 5 children, and none of those siblings look exactly alike because all of those siblings got a slightly different mixture of information from mom's egg and from dad's sperm. Okay, so after we've done that crossover in meiosis 1 is separating the sister chromatids from each other, so breaking up the homologous pairs, and then meiosis 2 actually splits the sister chromatids straight down the middle, just like in mitosis. So in meiosis 2, we're starting with the 2 cells that were the result of meiosis 1, each of these containing a mixture of new information. And at the end of meiosis 2, now we have 4 sperm or 4 eggs that are all different from each other. They all have now one set of genetic information, half the information needed to build a human being, but they've all got slightly different mixtures of information there. So, this one might have information to build brown hair, and this one might be blonde, and this one might be curly, and this one might be straight hair. So each of these different eggs and sperm can carry a slightly different combination of the information that was present in the parent cells. And so if we want to come back to this slide and just kind of summarize it here, remember mitosis, exact copies, meiosis, genetic diversity. Meiosis, half as much genetic information, so half of what you need to build a human, and a mixture of genetic information. A mixture that makes every single egg different from every other egg. A mixture that makes every single sperm different from every other sperm. Or if we want to bring it back around to this nice amoeba sisters image, again, exact copies versus having the genetic information and introducing genetic diversity here. And we're going to use this idea to kind of bridge ourselves into our next couple of topics, to bridge ourselves into the importance of that diversity to sustain life.